group that allows for proteins such as myoglobin and hemoglobin to bind to oxygen. It is a protoporphyrin ring that is made of 4 chiral rings linked together, with a central iron atom in the middle that is linked to the nitrogen atoms of the chiral rings. This process occurs mostly in the bone marrow and liver cells, and heme is also responsible for giving muscles and blood their distinctive red color. Heme synthesis occurs in both the mitochondria and the cytoplasm. The first step occurs within the mitochondria, where glycine and succinyl-CoA are reacted to form aminolevulinic acid. This reaction is catalyzed by aminolevulinic acid synthase, or ALAS. Afterwards, aminolevulinic acid is transported across the mitochondrial membrane to form porphobilinogen, and this reaction is catalyzed by ALA dehydratase. After porphobilinogen is formed, 4 of these are needed in order to react with porphobilinogen deaminase, and in this step, the amine groups are removed in order to form a linear tetrachiral. Afterwards, this linear structure cyclizes to form uroporphobilinogen 3. Afterwards, this is reacted with uroporphobilinogen decarboxylase, which removes the acetate groups in order to form copropyphobilinogen 3. This structure is then transported into the mitochondrial membrane in order to form protoporphyrin. Lastly, the enzyme ferroketolase is needed in order to add an iron to the center of the protoporphyrin ring, which gives us the final structure of heme.
The major regulatory and the rate-limiting step of the pathway corresponds to the reaction catalyzed by ALAS, which stands for ALA synthase. It is the first enzyme of the entire pathway, and also it is the first committed step to make heme. ALAS has 2 isozymes, which is ALAS1 and ALAS2. ALAS1 is expressed in all cell types, but ALAS2 is strictly expressed only in bone marrow. First, we're going to talk about the ALAS1 regulation. ALAS1 controls the heme production for basic cell function, and it is negatively feedback by heme to first, heme can be a co-repressor of the repressor of ALAS1 genes, which blocks the transcription from DNA to RNA. Next, ALAS1 mRNA could be blocked by heme, which stops the translation. Heme also has a function to destabilize ALAS1 mRNA, which causes the RNA to be degraded. Heme can also bind to ALAS precursors to prevent ALAS precursors from entering the mitochondria, which prevent the formation of ALAS synthase. ALAS2 is more essential than ALAS1 for heme synthesis, and it has a unique regulations with iron response element. When the iron concentration in our body is low, IRP will bind to IRE, which block the translation of ALAS to mRNA. When the iron concentration in our body is high, IRP will bind to iron and release IRE to let the translation proceed. In bone marrow, heme synthesis is almost always coordinates with globin synthesized, which means heme and globin are almost equal in concentration. However, when we get excess heme, Fe2 plus will be oxidized to Fe3 plus to form hematin, which hematin will either bind to ALAS2 precursors from letting it pass the mitochondrial membrane to form ALAS, or it can allosterically binds to ALAS2 to make a conformational change, which prevents ALAS2 from binding with its substrates.
I'm going to briefly discuss how heme synthesis plays a role in other metabolic pathways and processes. So as you've seen, succinyl-CoA and glycine are the two main reactants that are needed for heme synthesis because they're needed right at the beginning of the pathway. Now, if you look here, you see that both of those are located quite near the TCA cycle. So you're going to see that heme synthesis pathway ties in fairly closely here. And so much of the regulation upstream of that will have effects down towards heme regulation. Now, as a larger, more broad picture of how heme plays a role in the body, we can look and see that heme is used in multiple ways. For instance, it's incorporated into hemoglobin, myoglobin, and the cytochrome P450 enzymes, which is a superfamily of enzymes, some of which are often used in detoxification of barbiturates and alcohol in the body. So when we look then towards the overall regulation of heme and how it works in the body, we see that it has multiple positive regulators, which include alcohol, barbiturates, and hypoxia, which all makes sense considering that heme does play that role in detoxification of barbiturates, and then in hemoglobin and myoglobin, it is playing a role in oxygen transport. And then finally, a negative regulator of heme synthesis is glucose.